Since from this morning, she's been appointed associate professor in plant pathology. So my greatest congratulations from today. Today's session is going to point towards plant biology and especially redox regulation in plant biology. So it is quite an, I can't say I'm an expert on the topic, so I'm very curious about the talks. And right now already have a couple of questions of things which I'm mostly curious about, and confronting things about my field of research. I mostly work on humans, so it's what happens usually. So Professor Mäkinen she's a professor in plant pathology at Helsinki University. Her main focus research are the cellular and molecular biology of plant virus, and this has been the her biggest focus in the past 20 years. She's mostly working on the infection-associated molecular complexes, which are involved in virus translation, RNA silencing, and viral encapsulation. She's also investigating the metabolic pathways which are affected in the infection and the susceptibility genes which encode for proviral host factors. And as a matter of fact, the ultimate goal is the research and the identification of functional characterization of susceptibility factors that can create virus resistance in plants. So welcome again, Professor Mäkinen, the floor is yours. Okay.
Okay, so thank you very much. And we have here just a second, I will put just a pointer on.
Absolutely. So to the other people connected today, I ask you to wait for the questions for the end of the talks of the two speakers. So if you have any kind of questions, please post them on the chat and I'll just gather them at the end of the two talks.
Okay, so thank you for the introduction, and it's my great pleasure to speak today about the redox state changes in plant pathogen interactions. And here we have a nice view from the previous summer and from my potato field. What I'm going to talk today is first I will give a short introduction to ROS generation and quenching in plants, and then go into the plant immune responses against pathogens. And then, according to my research focus on viruses, I have selected two examples from virus-host interactions where redox plays a role. So first we go to this ROS generation and quenching in plants. The most common reactive oxygen species in plant cells are singlet oxygen and superoxide radical, then also hydrogen peroxidase and hydroxyl radicals. These are the main ROS species that we have. And they are mainly generated in different organelles like the plastids, chloroplasts, mitochondria, peroxisome, and also in the apoplastic space of the plant cells. In addition, there are some other locations where this ROS production is taking place. If you look at the ROS production in the apoplast, we have here the NADPH oxidase, which is called respiratory burst oxidase homolog D, which together with cell wall peroxidases are mainly taking care of this ROS production in the apoplastic space. And here the superoxide radical, which is formed, is already converted by superoxide dismutase to hydrogen hydrogen peroxidase. So then we get it transferred through the aquaporins into the cytoplasm, where it can also be converted by Fenton reaction to hydrogen radical. So we have perception of pathogen-associated molecular patterns on the surface of the plasma membrane. There are these PRR receptors that can activate this RP8OH and the ROS production. It is an enzyme that is penetrating the plasma membrane, and in the cytoplasm, this enzyme has a unstructured region that can be regulated by phosphorylation and also by binding to calcium. And these are then active, kind of taking care of the regulation of the activity of this enzyme. In chloroplasts, the molecular oxygen is converted to ROS during the normal reactions of photosynthesis, also photosynthesis and also photorespiration, and in mitochondria during the respiration reactions on the electron transport chain. So in chloroplasts, we have the production of superoxide radical from the photosystem 1 reactions, and also then singlet oxygen from the photosystem 2 reactions. In mitochondria, complexes 1 and 3 takes part into the production, particularly the superoxide radical via over-reduction of ubiquinone. Saya is going to talk more about these. And then in peroxisomes, during the photorespiration, we have the hydrogen peroxidase produced during conversion of glucolate to glyoxylate by this glyoxylate oxidase. And also the xanthine uric acid reaction with the xanthine oxidase is producing these radicals. So ROS production and quenching are in normal state, they are in balance. There is steady state equilibrium, and the ROS that has been produced can be also quenched by antioxidants. And ROS formation or this ROS elimination is either kind of the antioxidants for that are either enzymatic or non-enzymatic, and here we have the abbreviation of these enzymes that are typically taking part into these antioxidant reactions. We have the ascorbic acid and glutathione as main regulators from the non-enzymatic part, and there we have also the many phenolics and flavonoids, alkaloids, which are used to relieve the ROS in plants. Oxidative stress is then developing when the antioxidants, their capacity is over-exceeded by the ROS production. And similarly to other cell types, also in plants, of course, there are many consequences of the oxidative stress. And they include, for example, in plants, reduced photosynthesis, growth. There is defects in development and also crop yield. And many of the macromolecules of the cells are damaged. There are damages found in proteins, nucleic acids, lipids, pigments, and so on. And finally, when the ROS or the oxidative stress become really severe, there is cell death as the cause. If you look at the plant immune responses against pathogens, we have this so-called zigzag model that has been already known for years, and it really starts from the pathogen-triggered immunity, which takes place at the plasma membrane, where there are these plasma membrane pathogen recognition receptors, which are feeling or recognizing, or there is perception of the molecules derived from bacteria and fungi. But also during the previous years or recent years, it has been shown that even viruses are, although they are intracellular molecules or intracellular pathogens, they can also induce this PTI pathogen-triggered immunity. Then when this immunity is triggered, we have also effectors in the cytoplasm coming from these pathogens, and they can further enhance the immune reactions from the cytoplasmic side. So we have here the individual pathogen effectors, which can be recognized by the R genes, resistance genes that are in the cytoplasm. And this recognition is then also affecting this strength of the immune response. And when there is a successful effector-triggered immunity, which exceeds the threshold for HR, which is the hypersensitive response, then we get the hypersensitive response, which is a sort of programmed cell death in these plant, in these leaves of the plants, for example, or in the plants in general. The R genes code for these kind of the R genes encode for the proteins that are responsible for the pathogen recognition in the cytoplasm, and they are this kind of typical nucleotide binding leucine-rich repeat receptors that are either on the plasma membrane or in the cytoplasm. And these receptors are quite similar for many types of pathogens. They can recognize both fungi, they can recognize bacterial pathogens and also viral pathogens. And for example, few of these here of the intracellular receptors are against viruses. For example, this RX1 is against potato virus X. And this potato virus X is one of my examples that I'm going to talk today about. These are in fact, this picture is about the receptors that are recognizing viral effectors in the cytoplasm. And here, for example, this RX1, which is in the inactive form in the cytoplasm, after the recognition of the capsid protein of potato virus X, it is activated, taken to a nucleus, where it is starting the defense gene expression, which is then leading to these defense reactions and hypersensitive response. Many other receptors for different viruses. Here is one for tospoviruses, for example, and then we have two different ones against tobacco mosaic virus infection, for example, presented here. Some of them have been better characterized already, and some of them are still marked with question marks, how this signaling then goes to the nucleus after the recognition. The immune responses work through integrated signaling modules, and this pathogen recognition in the plant cells. For example, it leads to this hypersensitive reaction, which can be seen on these leaves here. This is after tobacco mosaic virus recognition. And this signaling goes through several second messengers like calcium. There are different ion fluxes which are induced. We have the reactive oxygen species that start to accumulate. There is also nitric oxidase and different lipid molecules that work as secondary messengers. And often these are, of course, also related to kinase cascades, like the mitogen activated MAP kinase pathway. The signals are perceived either at the cell surface or in the cytoplasm, in the nucleus or other organelles, and these all then contribute to effective response. One of the main molecules in the immune reactions is salicylic acid, which is upregulated immediately after pathogen infection by several in several ways. There is upregulation of the salicylic acid biosynthetic pathways in the chloroplasts, and this salicylic acid production is initiated by two different pathways: the isocharismatic pathway and also the phenylammonium lyase pathway. ROS and salicylic acid, they are kind of, the question was quite long, which one is upstream and which one is downstream, but it looks like that the ROS is both upstream and downstream from the salicylic acid upregulation. But anyway, this upregulation of salicylic acid is critical to trigger hypersensitive response and also systemic acquired resistance after this induction. Salicylic acid also plays an important role in accumulation of these defense-related metabolites in phenolic metabolites, and also it works in networks with several molecules, as we discussed already. But one important is this non-expressor of pathogenesis-related protein 1, which is NPR1. And this is really important for the transcriptional regulation, which salicylic acid is activating. We also kind of know that when, for example, the viruses are coming into the plant cells, there is injury on the plant cell wall and plant membrane, because the insects that are transmitting the viruses, they break the cell wall and plasma membrane, which triggers calcium influxes, and there is calmodulin binding transcription activator CAMTA3, which gets activated, and this activated CAMTA3 relieves suppression of the salicylic acid defense pathways. So there is induction of this salicylic acid dependent defense. So there is quite high or very complicated interactions in the plant cell during these ROS and salicylic acid dependent immune reactions. But this is kind of a schematic presentation, a simple one, telling that when we have ROS and salicylic acid activated and ROS produced in different subcellular localizations, we of course have from the ROS consequences to the antioxidants. For example, here the glutathione reduced and oxidized form is of importance, and salicylic acid from the other side is supporting this ratio is correct in the cells as far as it can. But at the same time, when this balance between ROS and antioxidants is regulated, we have also transcriptional reprogramming, and that is through this NPR1. In many cases, there is this NPR1 molecule that is in the cytoplasm first as an oligomer, and this oligomeric form is also having disulfide bridges that are kind of bridging the oligomers together. But when there is induction by salicylic acid, then NPR1 is transported to nucleus, and there it is interacting with TGGA transcription factors and Wernicke transcription factors, and this defense-related transcription starts. Also, salicylic acid plays important role in the HR, programmed cell death, in the induction in the infected plants, and it also regulates stomatal closure, which is a response to pathogen attack as well. For example, potivirus infection, and these potiviruses we are talking about later, it's inducing stomatal closure to also regulate the infection. So what I'm going to talk about is these potivirus diseases. First of all, we have the potiviruses, which forms a very big plant RNA virus group. They infect both monocots and dicots worldwide and practically in all climate conditions. So it's very important group of viruses, and especially potato virus Y infecting potato, and then also plumpox virus, which is infecting plum, apricot, peach, cherry, these stony fruit trees. They have been selected to be among the 10 most important plant viruses worldwide. They cause severe economic losses. The potato virus Y, and especially its necrotic tuber, its strain that is causing necrosis in tubers, is very important and difficult problem in the world at the moment. And this plumpox virus is causing a disease called Charcot, and Charcot disease is for example, causing that the non-mature fruits are dropping down from the trees, and also they are malformed, and this disease is really devastating, for example, in the southern parts of Europe. If we look at the potato virus Y infection, here we see in the picture how much it is, first of all, reducing the growth and also the yield. It's important always to use healthy seed potatoes to start to grow the potatoes. That is reducing this problem a lot, but it seems that it's really difficult anymore to control even in seed potato production in Europe. So the very northmost countries are still kind of having a little bit easier time to produce healthy seed potatoes. This virus is spreading through aphids. We have different host responses towards PBY. This, for example, this tuberculosis virus strain, depending on the genotype of the potato. We have some potato genotypes, for example, this Sante, that is expressing extreme resistance against PBY. And in these leaves, upon infection, there is no multiplication of the virus and hardly any symptoms. Then we have this hypersensitive reaction in resistant cultivars like Rival. And this can be seen as these necrotic lesions on the leaves. Some of the potatoes are tolerant, and in these leaves and in these plants, PBY can replicate, it can multiply, but at the same time, we don't see the symptoms on the leaves. And then we have some phenotypes that are sensitive and develop the normal disease, and we can see the symptoms. When the metabolites in these plants that are sensitive, this is this Igor genotype of potato, have been looked, and this was a study done in Slovenia in Kristina Kruden's laboratory. They infected these Igor potatoes with PBY-N strain, which is causing a mild infection, and also this PBY-NTN, which is causing an aggressive infection. And there were, of course, several metabolites that were affected. But for example, if we look at the ROS scavengers, what was observed was that first of all, there was, towards the end of the infection, on day 6 after the infection, induction of these metabolites. And in fact, first they were a bit reduced immediately after the infection, but then on day 3, in this N strain infected potato, where the mild infection develops, there was already increase in the ROS scavengers, not yet in these plants that were infected with this aggressive strain, but then on day 6 we had already upregulation of the ROS scavengers in these plants. And it is possible that this slow induction of this metabolite production, this ROS scavengers production, is giving the possibility for the virus to develop this aggressive infection compared to this milder strain. It was also studied how the transcriptional regulation is correlating with the metabolite profiles. And in fact, if you look, for example, this dihydroxyascorbate, which was upregulated by day 6, and also in the mild strain infected potatoes already by day 3, we see that in this mild strain infected plants, indeed the enzymes also that are required for this dihydroxyascorbate production, they are upregulated in the same manner as what we see from the metabolite profiles. Then if we look more in detail the chloroplast and plant immunity, first of all, the chloroplast-derived ROS play a pivotal role in establishing effective plant immunity. And quite often also similar, if we have the effectors in the cytoplasm, also the chloroplast ROS production can be affected by pathogen effectors that are directly and indirectly target targeting these chloroplast processes. And in this way, the pathogen can also try to suppress this chloroplast derived immunity. Now there are also the genetic markers for redox state changes in plants, and one of these redox sensitive markers is GFP that can be targeted also to chloroplast by putting the transition peptide into this reporter. And now this kind of transgenic potatoes were produced again in Christina Cruden's laboratory, and they have this reporter in the resistant rural potato background. And they did a study to look at the how the redox state is regulated during PVY infection in chloroplasts with these reporters. How these experiments were carried out is that we have here the potato leaf of this resistant cultivar, and we have here the lesions caused by the PVY infection. And now they have selected regions of interest so that this region of interest is overlapping with the cell death zone. And then the second region of interest is the one next to the first one, a little bit further away from this lesion area. Then as a control, they have used the area in the leaves that is from the healthy parts and also completely from healthy plants where there is no infection at all. And now we have here the images which are from the confocal microscopy, where we have this cell death zone marked. And here we have the resistant potato cultivar, and on the lower row we have a cultivar where which is again the resistance genotype, but it is a transgenic plant that carries this NIHG transgene, which is the salicylate hydroxylase that takes care of conversion of salicylic acid to catechol, and thereby it is in these plants this salicylic acid is depleted by this transgene. And now if this cell death zone and the cells that are in immediate vicinity of this zone are looked at, we can see that there is highly oxidized chloroplasts next to the lesion area. If we look, and that holds true in both types of these transgenic plants, also here where we don't have accumulation of salicylic acid to that extent. Then if this region that is a little bit further away from this cell death region is looked, we see that there is also some oxidized chloroplasts in these cells, but much less than here in the cell death region. And in the mock, there is of course the normal state. And if you look at the quantitation of these changes in this ratio with the excitation 405, which is for detection of oxidized form of the GFP, and then at this 488 nanometers, which is photoreduced form. Then we can see that there is significant differences compared to the controls, and also that there is higher oxidation level in the cell death zone than in these cells that are a little bit further away. And this is also true when the salicylic acid accumulation has been reduced by this NAHG transgene. There are also structures called stromols that are induced from the chloroplasts. They are tubules filled with the chloroplast-derived stroma, and they are induced by, for example, hypersensitive reaction. These stromols are likely involved in retrograde signaling towards the nucleus during pathogen infection, and as well they are involved in chloroplast movement in plant cells. When the induction of the stromols was also calculated and quantitated in this same study system, it's clear that the stromols are activated close to this cell death zone, and they are not that quickly activated in the salicylic acid depleted plants. Now, if we look at the conclusions from this study, so it's kind of clear that the increase in chloroplast redox state is linked to PCD signaling. And since this was the same both in these NAHG transgenic plants and the resistant plants, it is concluded that this PCD signaling is salicylic acid independent. These stromols are induced by cell death, which kind of is possibly kind of is putatively telling that there is a lot of signaling, and these stromols are induced especially in such cells where this PVY multiplication in front of infection is going on. That was tested in this paper. Signaling for HR conferred resistance to the virus is salicylic acid dependent, however, and this conclusion is from the cells with moderately oxidized chloroplasts, which have a lot of connections to epidermal cells with reduced chloroplasts. There is this high stromal connectivity reporting for active signaling, and there is a lot of transcriptional regulation also in these cells, which we did not look at the data. So this is proposing that these moderately oxidized cells are very active in cell-to-cell communication. Then I would like to take my second example of mixed infections, and I will start by telling that most of the very severe viral infections that are really devastating, for example, here, this maize field, they are often caused by 2 or more simultaneously infecting viruses. Here we have the maize lethal necrotic disease, which is caused by maize chlorotic mottle virus, and then one potuvirus. There are at least 3 different potuviruses that can work together with this maize chlorotic mottle virus, and all the maize plants are devastating or are kind of dead on this field, for example, here, whereas the potato is growing as the underzone plant here still. And one of the pairs that is causing this kind of synergistic infection is potex and potuvirus combination. it is causing a lot of transcriptome upregulation. There are many genes which are induced are functioning in responses to biotic stresses. Then there is also transcriptome downregulation. For example, chloroplast functions are downregulated. miRNA pathways are affected. For example, this copper zinc superoxide dismutase is regulated in these plants. There is severe ER stress because of overaccumulation of Potex virus protein P25. Accumulation of salicylic and jasminic acids, increased lipid peroxidation in cellular membranes, formation of superoxide radicals in chloroplast, and also dysregulation of antioxidant genes. But also increase activities of some of these antioxidant enzymes like catalases and superoxide dismutase. Here we see that if we do a single infection with PVX, which is the Potex virus part of this synergistic infection, or with PVY, we don't get as much of these cell death sites as in this mixed infection, which is here visualized by this coloration, dark coloration. Also, we can look at the superoxide radical production, and it is clearly increased in this mixed infection. We have been doing a phenotyping in our phenotyping facility, what happens in the growth and morphology of the double infected plants. And we see here, for example, that in this double infection, both the height and the surface area of the double infected plants is reduced significantly. Also the morphology, the compactness, how round this plant is. Here we have the control plant, we have singly infected PVA plant, we have a singly infected PVX plant, and then this co-infected plant. We see that also this compactness is decreasing, and at the same time the oval shape morphology is increased in these double infected plants. We can also see higher temperature in these upper systematically infected leaves of this mixed infection, and to some extent also in potivirus infection, which is kind of a consequence of most likely the stomatal closure. In PVX infection, the leaf temperature is just increasing to certain time point, which is 8 days per post-infection, but after that there seems to be a recovery from this increased temperature in these plants. If we look at the photosynthetic activity, there is this maximum quantum yield is reduced in this mixed infection to a certain extent. But now we face the problem in this phenotyping that we have some of the leaves also in this mixed infection that remain quite healthy because the virus does not enter into these leaves. They normally goes from the side of infection to the upper growing leaves mostly. And if we look at these pixels, how they look like in the control and also in these mixed infected plants, we see that if there is the normal cutoff for, or the cutoff for normal pixel values at 0.8, then we have much more of these let's say so that these stressed pixels, stressed cells in these double infected plants. And also in these radar plots, we can see that the operating efficiency, the photosynthetical efficiency, non-photochemical quenching, and many other parameters, they change in these double infections, which is marked here with this yellow shape in these radar plots. And especially we see this operating efficiency is reduced during the light adaptation of these plants. So there are clearly a lot of problems in the photosynthetic apparatus for these plants that are mixedly infected. Here we see how the gene expression of the virus is affected. We have here the infection with one Potex virus and one potuvirus. And if we look at this single infection with PVX compared to the double infection, we see in most of the time points this upregulation of gene expression in the locally infected leaves. But then when this infection is going systemic to the upper leaves, we see that the gene expression of PVX is going down. Also, we remember that the temperature was going down in these leaves, which is showing that there is less stress in those leaves. But then we see that the gene expression is really highly upregulated in this combination of two viruses. And it is specifically this potato virus X that benefits from this synergistic infection. It is known that this helper component proteinase, which is a potuviral protein, it is the silencing suppressor of potuviruses, RNA silencing suppressor. It's the gene that is, or the protein that is responsible for this upregulation of PVX gene expression. And if we look at the RNA silencing pathways in plants during virus infection, there is a silencing pathway that is this transcriptional gene silencing pathway for DNA viruses, and for the RNA viruses, we have this post-transcriptional gene silencing pathway that is activated. And as a consequence of catalytical activity of this dicer enzyme, we have small RNAs produced, and these small RNAs are stabilized by methylation, which is kind of carried out by HEN1 methyltransferase. And this HEN1 methyltransferase is using S-adenosyl methionine as the methionine donor in this reaction. Now this helper component proteinase has been shown to interact with HEN1, and in one of our previous studies we showed also that it interacts with S-adenosyl methionine synthetase and also this hydroxylase during the methionine, or which are important enzymes of the methionine cycle. So this potuvirus infection is affecting methionine cycle, and we have proposed that due to this interference, also HEN1 is depleted of the methyl donor, and the siRNA methylation and stabilization is therefore affected. Now, if we look at the what happens in these double infected plants, and especially if we start to play with these methionine cycle enzymes, that we for example, if we look at the SAMs or SAH Some of we silence them, then we see that also the glutathione levels, the reduced glutathione levels are affected. And especially if both of the some synthetase and hydroxylase are silenced and there is PVX infection, we have a lot of reduction in glutathione, the reduced form, and also definitely if the glutathione synthetase is at the same time silenced. And similarly, if we have PVX and HE-Pro expressed, not the potivirus infection, but only this RNA silencing suppressor, then we have a lot of reduction in the reduced glutathione. At the same time, when we have this reduction in the glutathione, we also see that the PVX gene expression is again upregulated. So we have hypothesized that we have a complex which is regulating the methionine cycle, and then when we have because of that difficulties in methionine production, the transsulfuration pathway coming from the cysteine here is taking place, and when the cysteine is targeted more towards methionine synthesis, the glutathione production is going down. So this was our hypothesis, and at the same time, the PVX gene expression is going up. Now that we made a metabolic profiling of these plants, we get a little bit further support to this hypothesis. What we can see here is that single infections by PVA and PVX are not that much affecting methionine synthesis. There is a bit of upregulation in PVX infection, and it's downregulation here at day 6, but otherwise in day 10 in the systemic leaves we don't see much of regulation of methionine amounts. But then when we have this double infection, there is serious reduction in the methionine in these plants. We also can see that there is serious reduction in glutathione amounts in the double infected plants in the systemic leaves at day 10. So now we are trying at the moment to analyze and make hypotheses out of this, how these metabolic pathways are affected by the double infection. What we can say that there is a reduction in the methylation capacity in these plants, as well that there is reduction in the glutathione reduced and oxidized form very clearly in these plants, which is one factor underlying this very severe stress and death of these plants. So as a conclusion, the descending ratio between reduced glutathione, which is important scavenger of ROS, and its oxidized form indicates that there is severe oxidative stress developing during this mixed infection, which might of course be extended maybe to other mixed infections, which can be studied. Then we were talking about this plumpox virus, which is a potivirus and causes this Charcot disease in stony fruit trees. There is a study where it has been shown that this plumpox virus infection induces oxidative stress in susceptible peach plants, and treatment of these plants with an artificial cysteine precursor, which is this L2-oxo-4-thiazolidine carboxylic acid, OTC. It is relieving the symptoms and stimulates the plant growth, and alleviates the virus-induced oxidative stress in peach. So what we can learn from these studies on the redox states and the metabolic pathways underlying the oxidative stress is that we, possibly, for example, this kind of environmentally safe compounds like this OTC could have potential to support healthy growth of virus-infected or even these co-infected plants, which I feel that this is an important finding and possible that it's important to study further if we can find this kind of compounds that could be used to take care that there is enough of food produced for the people of this world. So with this, I would like to thank everyone and for your attention and stop my presentation.
Okay, Christina, thank you. Very good presentation, very beautiful presentation. Already a couple of things, a couple of curiosity I'd like to address you later after the talk of Saija later. So now we pass to the second speaker of today's session, which is Professor Kangasjärvi. I'm really sorry if I botch your last names. I'm terrible at this, so I apologize in advance. So Professor Kangasjärvi is an associate professor in translational plant science at Helsinki University as well. She works mostly on plant light signaling and stress tolerance, and her lab focuses mostly on organelle dysfunctions and its effects on disease resistance and leaf senescence. They also aim at translating the new knowledge for improved indoor cultivation and post-harvest quality of leafy greens. Okay, so Saija, the floor is yours. As always, if there are any questions from the others, I invite you to wait for the end of the second talk. And so, Saija, the floor is yours.
Okay, thank you very much, and thank you very much for the invitation to this webinar series. So today I will tell you something about the mitochondrial dysfunction in plants. And here you see some photos from of our institute here in Helsinki, and it's not really so snowy yet, but we are coming to that season at the moment. Okay, my computer is okay. So I thought I'd give a short introduction to my lab first and then go deeper into the actual topic. So in my lab, we really look into fundamental basic mechanisms of plant stress tolerance. And then, as already kindly introduced, we then aim to translate these basic findings into practical applications. And for the research, why we do it is we think we are trying to answer to the current megatrends of climate change and urbanization. So with the climate change, we have already experienced exceptionally hot summers. Even here up in the north, we had an exceptionally hot, dry summer, whereas other parts of the globe are suffering really from flooding. So the complete opposite stress. And this warming in general and the humidity brings new pathogens and pests to areas where they didn't previously exist. So there's significant agricultural loss that Christina already nicely highlighted in her talk. But at the same time, with this urbanization, we need to get plant-based food in cities, and spoilage of food in the food chain is one big problem, and food waste is one global megatrend at the moment, which we should try to avoid. So solutions to this could, of course, be breeding more resistant cultivars of plants, crops. And one increasing trend actually at the moment is indoor cultivation. And the aim would then be to increase the nutritional quality of crops and the freshness and in the end consumption of plant-based food. And in this entirety, my lab aims for an improved understanding of plant stress, especially light signaling. And then we aim to apply this information to improve the nutritional value, but also flavor and shelf life of vegetables. And then if we look into more scientific terms, what this research is all about. So already for 15 years, my lab has been working on a crosstalk, molecular crosstalk between light acclimation and defense reactions in plants. And the rationale for this research comes from organellar interactions. So the current consensus is that when a plant senses the presence of a pathogen, now I have drawn this bacterial type pathogen in the apoplastic space, so outside of the plant cell, and plasma membrane resident pattern recognition receptor then first recognizes the presence of this pathogen and initiates a signaling cascade that regulates eventually gene expression in the nucleus to trigger these kind of defensive measures. But at the same time, there are protein interactions that upregulate and trigger the formation of reactive oxygen species, as Christina already told, first superoxide, which then dismutates to hydrogen peroxide in the apoplast or the extracellular space of the plant cell. And this initial reaction is performed by this respiratory burst oxidase homologs that are also present in different isoforms in plant cells. Now, in addition to these signals that are sent towards the nucleus, there's also quite an intimate crosstalk between these plasma membrane resident components and the organelles. And now that we are talking about plant cells, we have, in addition to mitochondria, also chloroplasts, which are the photosynthetic green organelles of plant cells. And they are really quite typically in membrane contact with each other. So there's a really intimate metabolic exchange between these organelles. And whichever of these organelles start going imbalanced and there's a redox imbalance and formation of reactive oxygen species, it's immediately reflected also to the other organelles. So chloroplasts, mitochondria, and peroxisomes, they are really closely connected. And sometimes in research, it's really hard to tell which organelle you're really looking at because of this intimate crosstalk. We do know, however, that these kind of disturbances, external stress conditions lead to formation of reactive oxygen species in the organelles that then trigger some signals, organellar retrograde signals that also reach to the nucleus and in their part also affect these gene functions in the nuclear genome. And as an outcome of these signals, some of these genes encode for organellar proteins and through anterograde signaling cascades send additional proteins back to the organelles in attempt to try to reach again a homeostatic status and balanced reactions in the organelles. But then there's also a more overall acclimation response that modifies the entire acclimation state of the whole plant. And these changes can range from changes in primary metabolism and the formation of so-called specialized metabolites that are also called secondary metabolites. All kinds of detoxification and quenching systems are activated, but sometimes also senescence or aging processes and cell death. And all these components have a huge impact on human nutrition. So through these kind of processes, environmental stress can really deteriorate the quality of our food. But some of these specialized metabolites are also healthy for us in our diet. For example, anthocyanins can have antioxidant activities in our diet, or the glucosinolates that are typical of brassica plants are also healthy in our diet. So there's a big impact to the metabolic state of the plant and how that affects us. And even if we think, at least we try to sell our research saying that this is very important, how these pathways are regulated is actually currently not well understood. So in this lecture now, we will be focusing on mitochondria and mitochondrial dysfunction. And now it is becoming increasingly clear that metabolic disturbances can cause mitochondrial dysfunction in plants. And this sensing can then trigger stress in the organelle. And the stress can trigger signals, and the signals can trigger responses. And it's not enough to trigger the defensive measures, but these should also be contained so that we don't exaggerate the defensive reactions. So there's only so much you can stress, so to say. And we are trying to understand how this works. Then if we zoom further into the mitochondria in this sequence of events, it's more or less the same thing again. So there is a stress. It can be biotic or abiotic, for example, ultraviolet light or too much light that can cause some kind of metabolic imbalance that leads to accumulation of reactive oxygen species. Now, from our view, this accumulation of ROS is seen as a transient event, and we will be discussing the antioxidant systems in just a moment. And it is actually currently not really clear what is the initiating signal in the mitochondria. So how the stress is sensed is actually currently not known. But we do know that the organelle send signals, and that one of the key signaling factors is a transcription factor, which in Arabidopsis thaliana, the model plant, is called ANAC17. And this is really considered the most significant transcription factor that then initiates a so-called mitochondrial dysfunction response that involves activation of genes that encode proteins that have functions as chaperones. They can be import components to organelles, respiratory components, metabolic pathway components, and growth and defense regulators. And all the stress response and cell death reactions occur during the acute phase of the stress condition. And then gradually later on, when the stress subsides, there should be acquired stress tolerance and recovery from the stress, and again, growth restoration. And already almost 10 years ago, Inge de Klerk showed that if you trigger this kind of mitochondrial dysfunction by different pharmacological approaches or by using different Arabidopsis thaliana mutants that are affected in mitochondrial functions, you get an activation of a set of genes that share a mitochondrial dysfunction motif in their promoter area. And this motif is essentially bound by the NAC17 transcription factor. So the NAC17 becomes activated. It's actually released from the ER membrane in response to these organellar stress responses. It enters the nucleus and binds this mitochondrial dysfunction motif, leading to activation of certain defense-related genes. And now I have marked in red some genes that are widely considered as marker genes of this mitochondrial dysfunction response. And they are not functionally really interconnected. One is a sulfotransferase that actually sulfonulates salicylic acid that Christina was just talking about. One is an alternative oxidase that I will introduce in just a second. And one is another ANAC type transcription factor NAC13. So these can be just considered as marker genes for activation of the mitochondrial dysfunction response in this presentation. So this is a busy slide, but the busyness illustrates the complexity of the mitochondrial pathways, and believe me, this is already a very simplified view to what happens inside the mitochondria, and now especially plant mitochondria. And before I walk you through the plant-specific aspects of this, I just want to highlight, using this picture I took from a colleague from Lund, is that the mitochondria are really tightly integrated in basically all cellular functions. So in metabolism, they are tightly connected with the photosynthetic organelles and through also photorespiration, which is a plant-specific process, and have a great impact on the cellular redox balance. And then through these activities basically affect the plant life from germination to seed production throughout the entire life cycle of a plant. And in addition to that, during the life cycle, the mitochondria greatly affect various kind of stress responses in response to both abiotic and biotic stress factors. So if we then zoom a bit into the mitochondria and raise some issues that are partially plant specific, if you look here, you see that the plant mitochondria have all the required or all the classical electron transfer components. So oxidative phosphorylation occurs through the complexes 1, 2, 3, and 4. But in addition to that, plants have some plant-specific components. And one of them is a so-called uncoupler protein, which can release protons from this matrix and which does not really result in a production or formation of ATP. It just releases the proton gradient. And another alternative electron transfer component is a so-called alternative oxidase. And this can quench excess electrons by transferring them to oxygen. And these two alternative pathways give quite a big flexibility to the mitochondrial electron transfer in plants. And if you remember from the previous picture, one of the marker genes for activation of the mitochondrial dysfunction response was this alternative oxidase 1A. It's a specific alternative oxidase isoform in Arabidopsis thaliana model plant. So this is one of the key regulatory components that significantly affect the mitochondrial redox state in plants, plant leaves at least. Then let's say when things go wrong, this actually happens during the normal electron transfer reactions that are taking place in the plant mitochondria all the time. But when there's some kind of an imbalanced state, for example, caused by an environmental stress condition, then especially the complexes 1 and 3 start transferring electrons to molecular oxygen, resulting in formation of superoxide in the mitochondria. And there's the superoxide dismutase is quite abundant and active in plant mitochondria, and it really rapidly leads to formation of hydrogen peroxide inside the mitochondria. And this can then be exported also to the cytosol. And now how this formation of ROS might be sensed in the mitochondria is one of the big question marks in the research field at the moment. Because it is also known that in addition to this SOD enzyme, there are also many tile-based and ascorbic acid-based antioxidant systems that quite efficiently quench the hydrogen peroxide, so that you don't really see under natural conditions this kind of prolonged formation of reactive oxygen species. But you can have a transient increase which can already be sensed, and it is known that these kind of signals arise. And to increase the ROS formation in experimental setups, we quite commonly use a compound called antimycin A, which somehow binds to the complex 3, inhibiting its function and leading to really enhanced formation of superoxide into plant mitochondria. And this is one very classical research tool that is widely used in plant research. So when we talk about mitochondrial dysfunction, it can be seen as both beneficial and harmful. So on one hand, activation of this kind of stress signaling can lead to an acclimated defense active state of the plant that can protect the plant from further environmental stresses. So it can induce this kind of cross tolerance, which is one phenomenon that's quite typical to plants. And here in the upper picture, for example, of figure, you can see that the Arabidopsis thaliana plants were treated with this antimycin A, and then a few days later were infected with different types of plant pathogens. And you can see that the antimycin A treatment, and likely then the consequent formation of reactive oxygen species and the signaling, resulted in increased resistance in all cases against these plant pathogens. And there's quite a lot of interest now towards understanding how this might be potentially used in crop breeding. Then there's another example that I find quite fascinating. So a group in South Korea found that radish taproots modulate the pithiness of the edible part of the root through a transcription factor NAC13. So another marker for this mitochondrial dysfunction response. And that this transcription factor actually triggers cell death in the taproot, and then it becomes kind of hard and woody and unpleasant in our mouth. So the sensory characteristics of these radish taproots are really unpleasant, whereas the other cultivar is much softer. And so the activation of mitochondrial dysfunction response in this pithy cultivar has quite a big effect and impact on the value, nutritional and also commercial value of this cultivar. But then the cost may be that this may be much more stress resistant. Another environmental condition that is quite important when it comes to mitochondrial functions and also the current climatic conditions and the climate change is flooding and submergence stress, where the plant is left or rendered underwater. And there has been quite a lot of research activity around this research area lately. And this example is from Jim Whelan's lab in Australia, where they found that mitochondrial signaling really is critical for flooding tolerance in the model plant Arabidopsis thaliana. So what they did was they just submerged the plants 10 centimeters underwater, and after a couple of days they collected samples and took some out from the water for recovery. So this desub means desubmergence. And then another time point that 5 days underwater, and then again recovery from the treatment. And here you see that the wild type Arabidopsis plants only started suffering 5 days after being submerged, some mitochondrial signaling mutants, notably an allele for this ANAC17 transcription factor, were really suffering and recovered very poorly from this treatment. And here with DAP staining, you just see how these two genotypes, so the signaling component mutants, really accumulated lots of reactive oxygen species, especially at the 5-day time point of the treatment. So there was a formation of ROS and really oxidative damage probably to these leaves, and they recovered very poorly. So the Wieland group then took some efforts to understand what kind of components might be involved in this response. And just like in us, when we suffer from hypoxia, these plants are starting to suffer hypoxia underwater, and there may be severe effects to mitochondrial components. And the Wieland lab then found that one of the targets for these transcriptional targets for this NAC17 transcription factor is aconitase 3, one of the key enzymes in the 3-carboxylic acid cycle. So this picture just shows that aconitase is a component, it's an enzyme in the citric acid cycle or 3-carboxylic acid cycle. And in Arabidopsis, we talk about aconitase 3 because Arabidopsis has 3 isoforms of this enzyme, and this particular one was identified in this screen. And so to understand how aconitase functions in the submergence stress, we had generated in my lab some mutants for aconitase 3 that we fished through some other way, and these were just happened to be available. And then the Wieland lab looked into their phenotypes in the submergence stress and found that the plants, Arabidopsis plants that were completely devoid of this aconitase 3 isoform, did not really tolerate the submergence stress and recovered very poorly during the desubmergence stress. Whereas plants overexpressing this aconitase isoform were quite tolerant to this flooding condition. And so we were then wondering what is the function of this aconitase, because somebody told me, well, it's just a metabolic enzyme. And I thought it can't just be a metabolic enzyme, there must be more to its function. And then we found that actually this Arabidopsis aconitase 3 carries a unique phosphorylation site that is not present in the other aconitase isoforms. And this is a bit peculiar. It's present in all kinds of cruciferous plants, including all kinds of cabbage and brassica plants and some conifers, cucumber, pineapple, but not across the kingdom. So there's something to this phosphorylation site we don't fully understand. And then we decided to take a little deeper look into this aconitase 3 phosphorylation, and what we found is that it is very stress responsive. So we chose to use ultraviolet irradiation to trigger an overall stress condition to the plant leaves, and then followed the abundance and phosphorylation of this aconitase isoform 24 hours later. So that the UVB treatment was a harsh treatment for 45 minutes. It's like a solarium for the plant. You would never experience that in the wild, but we used it as a research tool to trigger this kind of oxidative stress. And what we found in line with the Wieland identification of this aconitase 3 as a target for the stress-inducible transcription factor, also the protein abundance increases in response to stress, but also the phosphorylation level of aconitase 3 increases in response to stress. And this phosphorylation level increases more than the protein abundance. So we developed some targeted mass spectrometry methods to reliably quantify this phosphorylation event against the total protein abundance, and it was clear that the phosphorylation is quite significantly increased in stress conditions. And none of these regulatory actions took place in the ANAC17 knockout mutants. So when this key transcription factor is missing from a knockout mutant, we don't see the increase in the aconitase isoform abundance, and we don't see increase in the phosphorylation level either. So there's some interesting signaling going on. And essentially similar results were then obtained when mitochondrial ROS production was triggered by this antimycin A treatment. Again, the aconitase abundance increased, and the aconitase phosphorylation also quite significantly increased. So we were wondering how this kind of ROS-induced phosphorylation could impact plant stress tolerance. And so to look at it in a bit more detail, we generated another set of Arabidopsis aconitase mutants, where we placed the mutated versions of the aconitase enzyme after the aconitase gene promoter. So it was carrying its own gene promoter, and then there was a wild type version which had the normal serine 91 in this position. And then we created a phosphodeletion mutant where the serine was mutated into an alanine, and then another kind of phosphomimetic mutant where the serine was converted into aspartic acid. And these constructs were then transferred into the Arabidopsis aconitase 3 knockout mutant. So in the end, we had a wild type Arabidopsis, the aconitase 3 knockout line, and then these lines expressing the different variants of the aconitase 3 isoform. And in normal growth conditions, these plants were growing just fine. There was no evidence for accumulation of reactive oxygen species in any of the genotypes, and they were just very, very happily growing and reproducing as normally. But when we exposed these plants to stress, either UVB stress or antimycin A induced stress, we started seeing phenotypes in these lines. And here you see an experimental treatment with control plants. Again, all genotypes growing beautifully, no differences between them. But then here you see plants that were again exposed to a 45-minute UVB irradiation and then returned back to normal growth condition for 11 days to observe how they recover from this stress treatment. And here you see that the aconitase knockout, just like in the submergence stress, it didn't survive or it didn't recover well from the treatment. And the same was true for lines where this phosphorylation event was totally prevented by mutating the serine 91 into alanine. So this is a phosphodeletion version of the protein. But if you then look at the lines that were expressing the phosphomimetic version, they were doing a bit better than the aconitase knockout or the phosphodeletion line, but they didn't really recover to wild type levels. And the genotype that coped the best was again an aconitase overexpressor line, just like in the flooding stress. And here's the same thing quantified based on fresh weight. So you see that the aconitase 3 knockout and the phosphodeletion mutant are doing really badly. The phosphomimetic versions are a bit better, but the best surviving one is the aconitase overexpression. And this picture is very small, but you just have to take my word that all these constructs were targeted into mitochondria. So we are looking at something that is taking place inside plant mitochondria. And yeah, so here we were wondering about this phosphomimetic version. So how come it was performing worse than the aconitase overexpressor lines? And then we were thinking that it is really reversible phosphorylation that is required for the stress tolerance. So the protein must be able to undergo a phosphorylation, but a point will come when it also needs to be dephosphorylated, so that it is an on-off kind of regulatory action that is taking place. And then we also followed the phosphorylation level of the aconitase 3, both 24 hours after the stress and 11 days after the stress. And here you can see, note the scale, here is 1, while here is 3. So there is no longer any difference in the phosphorylation level of aconitase 3 11 days after the recovery period in wild-type plants. So aconitase 3 is really transiently phosphorylated. It is absolutely necessary for the plant to cope with the oxidative stress condition, but then it has to be dephosphorylated to allow full recovery of the plants. Now, we also saw some very peculiar phenotypes that we are currently working forward to fully understand what this aconitase is really doing and what its signaling functions inside the mitochondria might be. Because what was clear was that these phosphomimetic versions, so the lines that they're expressing the aspartic acid phosphomimetic version of the aconitase, were inducing these kind of mitochondrial dysfunction response markers already in the absence of stress. So these are just immunoplots showing that both the alternative oxidase abundance and sulfotransferase 12 abundance, so the marker protein abundance, increases in these phosphomimetic lines. And so we concluded then that this phosphorylation somehow triggers signals from the mitochondria that spreads to the nucleus, where it, through some so far unknown signaling activities, further activates. So it forms some kind of a positive feedback loop in the activation of this mitochondrial dysfunction response. And we are currently trying to understand how the reactive oxygen species signaling is changed in these aconitase lines and what these actual signaling mechanisms are. So we are hoping that, by using these genetic materials as a research tool, we will be able to get some understanding of how the stress is sensed inside the mitochondria. So is the mitochondria able to already sense the redox imbalances or even the accumulation of reactive oxygen species, or is it indirect through some metabolic interactions or accumulation of a given metabolite? And then in addition, what are all the signaling components and how do they interact with each other in this regulation? So there's a lot of unknowns underneath these arrows that we would like to fully understand how this system is operating. And when we finally understand a little better about the signaling and we see which of the components and mechanisms are conserved in crops and crop species, we can start considering strategies on testing these responses in breeding programs. And with these open questions, I would just like to thank the people in my lab and also these collaborators that have helped us quite tremendously to get this work done. And I thank you for your attention.
So, thank you, Saija. Beautiful presentation, absolutely. It was very, very interesting. I'm not usually used to talk about these arguments, and I find them absolutely compelling. There are also a couple of things which I find, how can I say, I didn't expect them to happen in particular conditions. So I'm going to see if there is some questions from the chat, but if there is not at the moment, I think I will start at first. I have a couple of questions for both of you. I would start with Christina. I think she's already online. Okay. I don't think I can hear you.
I'm here.
Okay, now I do. Sorry.
Yep.
So a couple of curiosity. I was looking at the first part of your presentation when you illustrated the hyper-resistant potato cultivars to the PVX viruses. Now, it's pretty, a curiosity. These kind of cultivars, are they natural occurring in nature, or are they selected through human intervention?
There is also, I mean, there are certain resistance genes in potato cultivars, natural resistance. There are also, of course, attempts to transfer the resistance genes, but there are few resistance genes that are in some of the cultivars. But
even without the intervention of
yes.
humans. And so how did it work? They possibly evolved alongside the viruses to be so much resistant.
Mm-hmm. Those natural resistance genes that we know, they may originate from the wild potatoes in Peru, but and they are more like these dominant resistance genes that I was today talking about. But then we have natural resistance, which are normally based on the susceptibility genes that are required by the virus infection, and these are more I would say more natural, or they are more abundantly maybe present naturally.
Mm-hmm. Okay. Also, another very quick curiosity. I was seeing at the overall effects of double infections in potato cultivars, the one from post-X and post-viruses, and I saw something which actually grasped my attention. I also took a screenshot. So you said that they downregulate the chloroplast functions, even though they upregulate the stress response genes, as a matter of fact. So is the downregulation overall of the chloroplast function is necessary to the infection, or is just a contingent effect of the virus? Because I would expect that in order for the viruses to be pathogenic, they would depress all the things which are related to the physiology of the plant. But strangely, you see stress response effects to be upregulated as a sign of the fact that the plant is trying to fight against the infection, but at the same time, you see chloroplast function, which is completely depressed. So is that maybe an adaptation? Is that a mechanism of infection, or is just a contingent effect of the infection per se?
Mm-hmm. The virus infection is dependent on the host, and therefore I think that it's all the time trying to find the best balance. And these double infections are definitely not very optimal in that sense that they are killing the hosts and killing that particular, maybe the whole field at the same time, so there can be 100% of yield loss. So I think that these single infections and in general the infections to start, they try to find the balance so that also the host can really support the viral functions and support the multiplication of the virus. And therefore, I think that at the same time, when there are these antiviral responses induced, there are also many types of proviral needs. So the virus has to take over the protein synthesis and quite a lot of the cellular membranes and energy metabolism also to reproduce. And I think that it's a kind of constant fight.
It's a game.
It's a game, like which one is kind of winning. So either the stress responses and defense responses, or the virus. And it can be also a question of timing, as we saw with this mild infection by PVY and the aggressive one, that in the mild infection, there was a quicker induction of these, for example, the ROS scavengers than in the aggressive infection. And also, you know, one really important part that I did not talk at all, not really just mentioned, that there is this RNA silencing, which also, I would say that is the main antiviral pathway against viruses, for example. So all these different R genes, RNA silencing, and at the same time the needs of the virus to multiply.
To not kill the host, of course.
Yes, you can understand that there is huge signaling going on.
We play against the current, basically.
Yeah.
We try to run against the current. And last question, this might seem very stupid, so I apologize in advance. About the metabolism of the glutathione, of the GSH, you said that if you knock out SAMs and SA hydrolases, in PVX infections, you see that the GSH production just plummets completely. Now, what's their role in physiological biology? I mean, if you knock them out without the infection, what do you see relatively to the GSH production? Are they induced as a sort of defense against PVX infections, or do they play a more physiological role? And when they are impaired experimentally during the PVX infection, do you just see GSH plummeting down further due to their lack of expression?
Mm-hmm. What is clear is that this methionine cycle is absolutely essential for the plant physiology. So if there is a knockout of those genes, it's clear that it's a huge stress for the plants, and somehow I think that the plant tries anyway to in which way it just can, it tries to keep the methionine cycle running. And I would say that the PVX infection, what is specific in that is that although the plant is suffering due to these silencing efforts, it's anyway beneficial for the PVX accumulation. And this is interesting, that what is behind that, why the PVX is doing better, and why its gene expression is upregulated in those plants. Mm-hmm. That is the open question.
Of course. There is also a question from the chat from Visnia Stepanik, and she's asking, have you measured status of secondary metabolites associated with stress, and are there some databases of a small molecular waste metabolites related to stress response effects in plants?
Mm-hmm. I'm sure that there are databases. I cannot give at the moment. I don't know, Sayalisa, do you know what is the link to some metabolite databases where the stress responsive metabolites are presented?
I am actually not aware of stress-related databases so much, but there are plant metabolite databases, but I don't have the links available now. But
yeah, I don't either have those now. But there is definitely studies on the secondary metabolites that are upregulated during the pathogen infections and other stress responses, and unfortunately either of us can now provide the link.
Okay, but there are databases. Anyway,
I have quite recently started to do this metabolite analysis, and we are currently preparing the manuscript on the findings of that I presented at the end of my presentation. But it's a good kind of question, and yeah.
We are starting to become dependent on, gathering information. So you can't basically work today without those. There is also another question from Marzia Reze. Can you comment towards the possibility of developing a protocol to increase oxidative stress in plants in order to increase antioxidant levels?
May I comment on this?
Well, of course.
Because that's essentially what we are doing at the moment.
It's an open session, so of course.
Yeah, so we are working on a collection of some 50 different kinds of kale cultivars, and we are using light, especially for this purpose, so that light through chloroplastic functions can trigger formation of reactive oxygen species and also reducing power in the chloroplasts, and that then triggers to an extent the formation of various antioxidants and in also species specific specialized metabolites. So yes,
so it can be done. So. I think it's very important because we hear every day talking about the fact that you should eat a lot of, for example, Brussels sprouts, brassica, because there are plenty in GSH. But of course, the natural occurring levels of GSH in those plants are important for the plants, but you should, eat tons of those in order to achieve a bona fide effect in animals. So, super GSH plants might be a thing in the future, or should be a thing in the future, I believe. As a matter of fact. Plus, I have a couple of questions for Saija too. I think we have, we have 5 more minutes, so give me time to bother you. I must say I was quite astonished by the thing that Aconite 3 is upregulated when you have a submerged plant, because as a TCA enzyme, I would expect that. Well, is it right to say that submerging conditions are, they closely relate to lack of oxygen, as a matter of fact. So upregulating an enzyme of the TCA cycle wouldn't be my first guess. So how does the two things, cope with each other? So you have not oxygen. Usually in animals, when you have like strokes, the first thing the cells do is downregulate the mitochondrial pathways to avoid the decoupling of the oxygen and the generation of a torrent in ROS. But in plants, if you're submerged, you upregulate aconitase. So does it mean that you'll also start trying to accelerate the TCA, or it's just a thing related to the phosphorylation of serine 91?
Oh, this is actually $1 million question. Thank you. It's a very relevant question. We don't really know, because the hypoxia and let's say the flooding stress is a really compound stress. So it could be hypoxia related stress, but it could also be some other metabolic rearrangements that are taking place. So it is currently not completely understood. But one peculiarity also of the plant TCA cycle is that it doesn't actually work as a cycle. So yeah. You get intermediates in and intermediates out.
You have a catalyst, basically.
It could just be a couple of reactions that are ongoing, and maybe the reducing agents are coming from degradation of metabolites under the flooding conditions. But then I didn't want to complicate the presentation too much, but aconitase could also have a moonlighting functions. So it could also turn into a regulatory protein, and in mammalian cells, it's also known to act as in response to iron stress as a regulator of gene functions. And these are very poorly understood in plants, and so it could be that it's actually not necessarily a metabolic function. It could be a
signaling function. Another thing which was wonderful, the phosphomimetic mutation of the serine 91 on the aconitase 3, is it known any structural basis behind this? The fact that if you mutate the serine with aspartate, you not only you simulate the phosphorylation, but also you get a protein which is most effective than the wild type genotype. Is there any structural basis, but maybe, I don't know, ionic couple which is restored or stabilized?
This is another question where I don't really have an answer, because the N-terminus of this aconitase enzyme is somehow flip-flop outside of the core structure of the enzyme.
Right.
And it doesn't have any obvious structural effect. And we also looked into complex formation, so how it's bound into protein complexes, and we didn't see any differences regardless of the phosphorylation status or the phosphomimetic and phosphodeletion versions. So we don't have any evidence that it would affect mitochondrial import or complex formation. But we do have some indications that it does affect the enzymatic activity of aconitase.
Right.
But then whether it somehow affects the moonlighting functions
or the proper
like this
role.
is something we are currently trying to understand what's going on.
Okay, wonderful. Okay, I think I'm looking for other questions on the chat. I don't think. Okay, I think we've exhausted them all. Okay, I think we are fine. I would like to thank you for this wonderful presentation. It was my utmost pleasure to have you here at the Redox Webinar Series, and hope to hear from you in the future.
Okay, thank you very much.
Thank you.
You're very welcome. Okay, have a good day, or good night actually, since it's quite late.
Yep.
Thank you for your work. Yep. Have a good one.
Thank you. Bye-bye.
Bye.
Bye-bye. Thank you. Okay, with this we close the session of the Redox webinar. Thanks to all those which had some time to connect and stay with us, and I'll see you at the next appointment, which is going to be in December, if I'm not mistaken. So have a good one. Good night.